You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is April 15th, 2024. Our topic today is Abbas Chapter 13, Effector Mechanisms of Humoral Immunity, presented by Dr. Nikita Raje. She is an Associate Professor of Internal Medicine Pediatrics and is the Chief in the Section of Allergy Immunology at Children's Mercy Hospital.
Talking about effector mechanisms of humoral immunity from Abbas, that's chapter 13. Again, I'm Nikita Raje from Children's Mercy Kansas City and University of Missouri Kansas City. That's my email in case you need to send me any questions or thoughts on these presentations. Today is April 15. Wow, April 15, 2024. Already half the month is gone. So let's see if I can move this presentation somehow. All right, here are my disclosures, nothing relevant to the talk today. Our objective for today are to expand knowledge about humoral immunity. And humoral immunity works through neutralization of toxins, opsonization, and phagocytosis and complement system. So we'll talk about each of those. Few questions. Which of the following functions of antibodies does not require Fc receptors specific for IgG? Children with C3 deficiency are at high risk for serious bacterial and fungal infections. Which of the following explains the importance of C3 in defense against these organisms? So lots of choices there. We'll come back to these. All of the following are accurate statements about neonatal immunity except. And a 15-year-old boy with a history of multiple episodes of severe abdominal pain is found to have evidence of intravascular hemolysis, including hemoglobinuria and an elevated retic count. Further workup included flow cytometry for RBCs and showed a lack of cell surface GPI. Linked proteins. A diagnosis of PNH was made. What is the basis of loss of RBCs in this patient? All right. Give me 1 second here. Trying to see. I'm gonna make sure that I have this right. Sorry, give me 1 second here. Can you guys see my screen yet?
I can't see it.
Okay, how about now?
Yes, I see it.
Okay, all right. All right, sorry, that was just a technical issue. All right, so let's get started. Let's talk about humoral immunity. It's mediated by secreted antibodies, as we know, and defends against extracellular microbes and microbial toxins. What this means is virus is an intracellular microbe, but when that virus is outside the cell, the humoral immunity can still affect it, right? So it's the extracellular phase of microbes, not a particular microbe that we are talking about. When the microbe is in the extracellular phase or in that space, that's when the humoral immunity can target those microbes. When that microbe is intracellular, the humoral immunity is not an effective response to that phase of the microbe. Defects result in increased susceptibility to infections with bacteria, fungi, viruses, and humoral immunity can mediate tissue injury in atopic individuals or autoimmune disorders. So we know that vaccines can produce humoral immunity. And so for some of these vaccines, the mechanism can be different. So for example, for polio, the virus is neutralized by the immunoglobulins, IgG, IgA, tetanus and diphtheria toxoids. Again, neutralization works great. Same for hepatitis A or B. It's a neutralization of the virus. Compared to pneumococcal or Hib or Neisseria, the vaccines are conjugates. Those are composed of bacterial capsular polysaccharide that's attached to a carrier protein. And in this case, there is opsonization and phagocytosis that's mediated by immunoglobulins, either directly or secondary to the complement activation. So rather than neutralization, it uses opsonization and phagocytosis. So thinking of the effector functions of these B cells and antibodies, we know that the antibodies act at sites that are distant from where they are produced, right? They come from short-lived. So the effector cell or form of B cells is plasma cells. These plasma cells can be short-lived or long-lived. So plasma cells can be in the bone marrow or spleen or lymph nodes. They can also be in mucosal tissues. And sometimes we know primary lymphoid tissue is bone marrow. Secondary lymphoid tissue can be spleen or lymph nodes or mucosal tissue. But there's a concept of tertiary lymphoid organs, which means when there is chronic inflammation going on, that site turns into a lymphoid kind of, we call it tertiary lymphoid organ, which means there is a lot of lymphoid activity. And there are plasma cells as well that can produce immunoglobulins. So that the major chunk of plasma cells are in the bone marrow where they produce the antibodies, but there are other places where plasma cells can act. All right, and the effector function is mediated by the immunoglobulins using their heavy chain constant regions or FC region, right? And so the FC region varies by the isotypes, and so the effector function also varies by isotype. So IgG, M, A, E, all of them have different functions based on the FC region that they have. Now we know these, in this figure, we see that the B cell producing antibodies can work through neutralization, phagocytosis and opsonization, antibody dependent cellular cytotoxicity, or complement activation. And the complement activation then can lead to phagocytosis, inflammation, and lysis of microbes. So based on the isotypes, these are the different isotypes we know of, and there are various functions for each of them. IgG, as you see, has several different ways of action, including opsonization, activation of classical complement pathway. It helps with the aid of NK cells, and it provides antibody-dependent cellular cytotoxicity. It also provides negative feedback to B cell, so causes inhibition of B cell activation. And it provides neonatal immunity. That's because of transfer of maternal antibodies across the placenta and also across the gut. IgM is important in classical pathway activation of complement. IgA helps with mucosal immunity. I think of IgA as border security. It sits there along the mucosal lining, either GI tract, respiratory tract, and whenever there is a microbe or a toxin that's trying to enter the body, it basically attaches to it and pushes it away. It basically neutralizes those threats to immunity. IgE causes mast cell degranulation and helps with eosinophil-mediated defense against some of these bugs. So let's talk about each of these mechanisms that the antibodies use for their function. The first is neutralization. How do antibodies use neutralization in their function? The first one is steric hindrance, which means the antibody attaches to the microbe in a way that it cannot bind to the cell that it uses typically to enter the body. For example, in case of influenza, it enters the body using respiratory epithelial cells, but if the antibodies bind to that area of influenza virus, that it cannot use that part to attach to the respiratory cells. Or gram-negative bacteria that have pili, they use those pili for their pathogenesis, but if the antibodies bind to that area, they cannot function in the same way. Same way the tetanus toxin or the diphtheria toxin, the antibodies can just attach to those to those toxins to prevent the action of the toxin. That's steric hindrance. There's also something called allosteric hindrance or effect, which means that the antibody doesn't really bind to that area that attaches to the cell. So it's that microbial part of that microbial surface is free, but because it attaches to the microbe in a way that causes conformational change in the microbial surface, and that means that even though physically it's not attached to that part, it causes conformational change so that the microbe doesn't really bind to or enter the system as it would otherwise. And then this function is mediated by the FAB region. Unlike other functions of antibodies that are related to FC region, neutralization is a function that it performs using FAB region. So all isotypes can be part of that. IgG is using neutralization mostly in blood, IgA mostly in the mucosal area, and unfortunately some of the microbes will mutate the genes encoding the surface antigens so that they can evade the neutralizing antibodies. The best example of that is influenza virus that changes its strain pretty much every year. We see different strains that come out, and they are doing that so that they can evade the neutralizing antibodies, right? So neutralization can be from steric hindrance or allosteric effect. Let's talk about opsonization and phagocytosis. We know opsonize means to decorate, which means to kind of tag that bug so that it's ready for phagocytosis, right, to be consumed by the phagocytes. So it's a process of coating the particles to promote phagocytosis. The substances that perform this function are called opsonins, and that can be antibodies or complement fragments. The best opsonins in terms of antibodies are IgG1 and IgG3. And so once those microbes are opsonized, those antibodies have their Fc region that can bind to Fc receptors, right? Fc receptors are on several different cells. Phagocytes have this particular Fc receptor called Fc gamma R1. Gamma, which means that it's going to bind to IgG isotype. So Fc gamma R1 is the most important receptor for phagocytosis of opsonized particles. So here in the figure you can see here is the microbe. It gets opsonized by IgG antibody. Now it's because it's ready for consumption, this phagocyte is gonna attach to that antibody using the Fc region binding to Fc receptor, so that in terms of that, now there will be a activating signal so that microbe that's attached to those IgG are going to be causing a pouch to be formed so that it will phagocytose that microbe. And so that microbe is going to be inside the phagocyte in phagolysosome, right? So that's how opsonization and phagocytosis works. Opsonization is provided by immunoglobulins or complement fragments. So we talked about Fc receptor, FcRn, gamma R1, which is the most important for phagocytosis, but there are several Fc receptors on various cells that bind to the Fc region of various antibodies. We know there are different Fc isotypes based on Fc region, so there are different receptors for different isotypes. So for IgG, it's gamma receptors. For IgE, it's epsilon receptors. And for IgA, there is alpha receptors, right? But these receptors have different subtypes. So Fc gamma is Fc gamma R1, R2, A, R2B, R2C, and R3. A and R3B, right? So these are all gamma receptors. As you see, they are numbered, CD numbers that are allotted to each of them, and you have to memorize these. And really simple way of memorizing these is it's the reverse of the number. The number reduces as the R number goes up. So R1 is CD64. Cut that into half, you have CD32, that's R2, and then cut that into half, it's CD16, and that's your R3, right? So gamma receptors, Fc gamma receptors are numbered CD64, CD32, and CD16. Really important thing to remember. For example, the CD16 is important for antibody-dependent cellular cytotoxicity with the help of NK cells. So that's Fc gamma R3A. There's also R2B that's important, CD32, which is the inhibitory Fc receptor that provides feedback inhibition for B cells. And as you see, it's on other cells as well, but it provides negative feedback. The most important activating receptor for gamma is Fc gamma R1, important for phagocytosis, as we just looked at. Also R2A, that's also important for phagocytosis. R2C, similar, and then R3B is CD16. It also helps somewhat with phagocytosis. Fc epsilon R1 and R2, there are two, but the R1 is the one most important, and activation helps with mast cell activation and degranulation. And then Fc alpha R is on for IgA. It binds to IgA. These receptors are present on various cells like neutrophils, eosinophils, monocytes. So let's look at these Fc gamma receptors now. We are talking about just the receptors that bind to IgG, right? Neither E or A. We are talking about IgG, and there are three groups, as we saw: R1, R2, and R3. And these receptors, most of them are activating. So at the cytoplasmic tail of that is your ITAM, because it's activating receptors, whereas R2B is the inhibitory one that has ITIM on its cytoplasmic tail. These have various chains. As you see, structures are a little different from each other, and all of them do have this alpha chain that binds to the IgG, right? So that part is the same that binds to the IgG. Fc gamma R1, that's our CD64. It binds to IgG1 and IgG3 subclasses with really high affinity, and the alpha chain is linked to a two gamma chains with ITAM motif. Did it work? Did it work? Yes, R2. Fc gamma R2 is CD32. Again, we saw that R2B has the ITIM, the other two have ITAMs. And CD16 is a low affinity receptor. It has, again, A and B forms, and pretty much, provides low affinity activation for binding to IgG. Another one that I wanted to talk about is FcRn. FcRn, N stands for neonatal receptor. Fc receptor that is used for passive immunity. It helps with binding to IgG and keeping it from degradation for a longer time. That's how the immunoglobulin G gets longer half-life compared to other immunoglobulins. Now, FcRn has affinity for acidic environment, and its structure is similar to MHC class 1 molecules. So there is a transmembrane heavy chain that binds to a beta-2 microglobulin, and as I said, it provides with passive immunity, so it can help transfer IgG across placenta. Both IgA and IgG that's secreted in breast milk, and it gives higher or longer half-life to the immunoglobulins because it prevents catabolism or breakdown of IgG. So how does that work? So here, let's look at this. This is baby circulation, this is mom's circulation, this is IgG in mom's blood that is taken up by the placenta in these small endosomes. And in the early endosome, when the immunoglobulin comes in, it basically in acidic environment can bind to the FcRn. So the acidified endosome will have FcRn bound to the immunoglobulin in this endosome. So that prevents it from breakdown. When it goes across that, comes close to the fetal circulation, this acidic endosome, as at normal pH, will release the immunoglobulin from the FcRn. So when it enters, then it enters the fetal circulation, and the immunoglobulin can go across that that barrier of placenta.
Dr.
So that's
Raje,
your.
so the IgG, it's binding to a macrophage in the endosome, right? Or it's binding to what type
It's
of
a
cell?
syncytial trophoblast. It's
Oh, okay.
placental cell.
Okay.
In that placental cell, there are these, because in that area, there might be acidic pH. So then as it's taken up, that endosome is able, the FcRn is able to attach to that immunoglobulin. But as the pH neutralizes, the FcRn can release the immunoglobulin. That happens not only in the placenta, but also in the gut. And that's how the immunoglobulin overall can get longer half-life, because that immunoglobulin is not broken down. So yes, here this is placental cell, but it could be other cells like macrophages that will not break down that immunoglobulin.
Okay, thank you.
Mm-hmm. So antibody-dependent cytotoxicity, antibodies bind to the antigen that's expressed on the surface of a cell. So here is a cell that has surface antigen that got kind of opsonized by immunoglobulin. That cell is an infected cell or a damaged cell that NK cell is targeting. NK cell has Fc receptor R2A or CD16 on it that it will use to bind to IgG bound to that damaged cell. So that damaged cell will undergo apoptosis. All right, when we talk about IgE, typically we are talking about worms or helminths that are too big for phagocytosis. So the same mechanism that's used by IgG may not work. That worm that's attached to an IgG will not be able to be phagocytosed by macrophages or other phagocytes just because they are too big. Instead, what happens is the IgE can bind to those worms, right? And now that IgE is going to bind to the cells using their Fc epsilon R1 or Fc epsilon receptor, right? For example, eosinophils or mast cells, they have Fc epsilon receptors on their surface that can bind to IgE. IgE bound to that bug will lead to degranulation of, or lead to activation of eosinophils, degranulation of some of these mast cells, or the eosinophils will release granules, and those granules that have enzymatic or toxic activity will be able to kill that helminth. All right, moving on. Now we'll talk about the complement system. This is one of the effector mechanism used by humoral immunity and also innate immunity. It helps with, it promotes phagocytosis of microbes, right? And then, so it opsonizes and helps with phagocytosis. It also causes inflammation, and then it leads to lysis of the microbes using the membrane attack complex. So complement proteins are small proteins. They are called zymogens. They are present in the plasma, and when activated, they can bind to each other to form bigger complexes. These are complement proteins are activated by various ways. It could be directly by microbes or antibodies, and for one of the pathways, it's just, it's just in the active form. So overall, the activation leads to sequential proteolysis of the proteins, generate bigger complexes or that have enzymatic activity and proteolytic activity. So that's the reason they are called zymogens. They acquire these proteolytic activity or properties. And the complement proteins are activated after their attachment to microbial cell surface or antibodies that are bound to those microbial surface or to the apoptotic bodies. Activation is inhibited by some regulatory proteins that are present on normal host cells but absent from microbes. That's how our host cells are protected from being killed by the complement system. So we know complement system has three different pathways: classical pathway, alternative pathway, lectin pathway. Alternative pathway is your default pathway, which means the pathway is constantly releasing complement fragments. If they do get attached to a microbe, it will be activated. If not, it's just continuously slow. There is a little bit of tick over. And then lectin pathway has that plasma lectin, and the classical pathway is the pathway where the antibodies are involved. So they are activated by antigen-antibody complex. Overall, if you see here in this figure on this left panel, these are the common steps to all three pathways. There is binding of complement protein to microbial surface or antibody. And then there is formation of something called C3 convertase, which helps in cleavage of C3. And then there is formation of C5 convertase, and because of which there is cleavage of C5. This process is different from each pathway. Once that C5 makes C5A and C5B, the final common pathway starts, which is common to all three pathways. So the big ways these three pathways are different are in how they form C3 convertase and C5 convertase. Let's talk about alternative pathway. Here are some of the proteins that take part in that: C3, factor B, factor D, propyridine. Factor B is a protease that helps in, it's the active enzyme of C3 and C5 convertase. And factor D helps in cleaving factor B when it's bound to C3b. So again, an important enzyme. It is a protease that takes part in that initial activation. Propyridine actually stabilizes C3 convertase on the microbial surfaces. So again, plays a big role there. So here's our alternative pathway. C3 is constantly being released, and the intact C3 piece has something called a thioester group. So chemically, it has this bond in it that's cleaved when there is cleavage of C3. So when we say C3 is cleaved, this bond is broken. So it leads to formation of two fragments, C3A, smaller fragment, and a big C3B fragment. C3A leads to maybe, just inflammation, but the C3B is the one that has the broken thioester group, but that's accessible. When it's in the fluid phase, if it doesn't come across any microbe, that's going to be neutralized or inactivated by hydrolysis, compared to if there is a microbe in that area, it is able to attach to that thioester bond. So when C3b is bound to a microbial surface, it becomes a binding site for factor B. As the factor B gets attached, factor D comes along in helping, and that part is stabilized by propyridine. And so C3 continues to be cleaved to C3A and C3B. That C3B bound to the B, factor B, is the one that's called C3 convertase. So C3B, BB is called a C3 convertase that helps in cleaving C3 very efficiently. And once that continues to do that, another piece of C3b is bound to the C3 convertase to form C5 convertase. And then finally, that starts cleaving C5 very efficiently. So again, C3 cleaves to C3A, C3b. C3b binds to the B part of, like, the fragment of factor B that's cleaved by factor D. And so factor B has two fragments, BA and BB, and that BB binds to our C3b to form C3 convertase, and then another piece of C3b binds to that complex to form C5 convertase. All right, now let's move on to talk about the classical pathway, how that gets activated. The proteins of classical pathway are C1, CQ, C1R, C1S, C4, C2, and C3. C2 has the serine protease activity that is important for making that convertase. In the C1, it's a complex of two molecules of R, two molecules of S, and C1Q. C1Q is binds to the Fc portion of the antibodies, and pretty much C1 is the one that activates the classical pathway. The enzymatic activity is in R and S parts that help to cleave some of these proteins. So here is the structure C1Q, and that those small two molecules of S and two molecules of R sit in that structure of C1Q. That's the whole C1 molecule, right? This C1 can bind to antibodies, as you see in this figure, and it can bind to multiple of the antibodies. And then because of that, the C1R and C1S serine proteases get activated. That helps in cleaving when more than one or more than two Fc regions are bound to that C1Q. It gets activated. The Fc regions of the soluble IgM are not really accessible. So here you see this molecule here for IgG. When there are two molecules that are bound to it, it can attach and cause complement activation. When the IgM is bound to a surface, it can bind to, because it has multiple areas that it can attach to. C1Q can bind to IgM, but it has to be attached to a surface, a microbial surface, to be able to target that C1Q. Once it's attached, there will be complement activation. C4 is similar to C3. C1S helps to cleave C4 to C4A, C4B, and C4B contains the thioester bond that we talked about. It remains with the antigen-antibody complex. So C4 is cleaved to C4B that attaches to the microbial surface. C2 complexes with that C4B. And it's the C2A that binds to that C4B, and that C4B, C2A is your C3 convertase, which means now it's going to start cleaving C3 very efficiently. Again, another piece of C3B can bind to this C3 convertase to form a C5 convertase that helps to cleave multiple molecules of C5. Lectin pathway has very similar structure to C1, but the proteins are named differently, mannose binding lectin, and also has that instead of C1R and C1S molecules, there is something called MASP1, MASP2, MASP3. They have the enzymatic activity. Similar to that, there is ficolin that has also similar structure to MBL. So here's your MBL, very similar structure to C1, and then bound to two molecules of MASP1 and MASP2 that have the enzymatic activity. Similarly, ficolin has ficolin protein along with MASP1, MASP2, right? These have the enzyme, those small molecule, those proteins have the serine protease activity. So each of these will bind to particular type of microbe. So any microbe that has mannose sugar in it, the MBL can bind to. Anything that has any acetyl glucosamine on it will bind to ficolin. All right, so the late stage of complement activation includes these proteins C5, 6, 7, 8, and 9. So let's see what happens there. Now we have any of these pathways, alternative, classical, or MBL. The end result is that there is a C5 convertase that's cleaving C5 to C5A and C5B. as a lot of that C5 accumulates, the C5b continues to stay on the microbial surface. C6 and C7 then come and attach to that C5b and becomes a receptor for C8. C8 is a trimer, and one chain inserts into the membrane, other binds to C5b and C6 and C7 complex. Once that happens, basically the final component, which is C9, comes in and gets inserted into that complex. Multiple molecules of C9 or poly C9 comes and attaches to form that hole in the cell. That's your membrane attack complex, including C5b, C6, C7, C8, and C9. Complement receptors are 4 types: CR1, CR2, CR3, CR4. You have to remember these. CR2 is really important. CD21, that's part of the co-receptor for B cell that we talked about. It's also a receptor for EBV, so really important to remember that one. CR1 is your complement receptor that's important for phagocytosis. CR3 and CR4 are important because their CD markers, look at them, CD11B, CD18, or CD11C and CD18. That's important to remember. Those are your integrins, and if there is a defect in CD18 causing leukocyte adhesion defect, that can lead to defect in complement receptors CR3 and CR4. For obvious reasons, right? So remember these 4 complement receptors: CD35, CD21, CD11B, CD18, or CD11C and CD18. All of those are complement receptors that can bind to complement fragments. There are some other molecules that help in regulating the complement activation. They are called regulators. They include some of these factors. So C1 inhibitor, right? C1 esterase inhibitor, we have heard of that. Factor I, factor H, C4 binding protein that binds to C4b and displaces the C2, membrane cofactor protein or CD46, decay accelerating factor or DAF, CD59. Each of these have a particular role. Membrane complex, a cofactor protein is a cofactor factor I that helps in cleaving C3b, the active parts of C3b and C4b, and inactivate them. Factor I is a serine protease and cleaves those with the help of the membrane cofactor protein. Decay accelerating factor displaces C2a from C4b and bb from 3b, so it dissociates C3 convertase. And CD59, even if all those steps go through, CD59 is important to block the C9 binding and prevention of membrane attack complex. Factor H is a cofactor for factor I mediated cleavage of C3b.
Raje, just a quick question. So all these factors that are just floating around in the blood, they're always there. They're just made by the liver. Is there anything that upregulates these, or are they just... I'm just curious. Like, it's interesting that there's so many ways to stop complement activation, so it just makes me wonder why that
And
is.
it's important, right? Yeah, they're always there. They're always present because that's how all our surfaces, all the host cells are not damaged by complement, because there is otherwise always C3 that's cleaving from alternative pathway that will grab our cells and kill them. But that doesn't happen because of all these factors that are present. And if there is a defect in one of these, such as in DAF, there is a problem, right? So yeah, so they are always present. So I think the next slide will tell you what happens if one of them is defective. Like this one tells you how if C1 esterase inhibitor is defective, we know that deficiency causes HAE or hereditary angioneurotic edema, right? So this is an inhibitor, serine protease inhibitor, which targets C1R2 and S2, right? R1, R2 and S2. So this is C1q, and the C1q inhibitor is supposed to prevent that activity, and if that's defective, it pretty much stops the activation of classical pathway, but if it's defective, it leads to edema. Inhibition of C3 convertase, so C3b and 4b can bind membrane proteins, so MCP, CR1, decay accelerating factor, factor H. All of these are making sure that the complement does not stay active when it's not supposed to. But if there is defect in the decay accelerating factor, for example, there will be paroxysmal nocturnal hematuria. Factor I is a serine protease. With the help of membrane cofactor protein, factor H, C4BP, CR1, all of these are cofactors for factor I, and factor I helps in mediating cleavage of C3b and C4b so that they are converted to inactive products. However, defect in this will lead to atypical HUS as well as increased infections. So factor I or factor H defect leads to atypical HUS but if factor I is defective, there are increased infections. Inhibition of MAC formation, CD59, S protein help in preventing formation of membrane attack complex, or S protein helps in preventing the insertion into the membrane. We talked about the effector functions of complement. They help in complement helping in lysis, right? By membrane attack complex, it helps in opsonization. The smaller fragments C3a, C5a, all of them cause inflammation and also lead to also called anaphylatoxins. And then the complement helps in clearance of immune complexes. So if there is a defect of complement, there are these immune complexes that continue to stay that cause autoimmunity in complement defects. All right, we talked about the complement-mediated cytotoxicity, where the membrane attack complex makes a hole in the microbe and causes the lysis of the cell. Opsonization and phagocytosis. The C3b is bound to the microbe that leads to the opsonization and helps target the phagocytes, are given the signal so that they can target that microbe. Inflammation is caused by these smaller fragments that bring in other leukocytes. So they recruit the leukocytes from the circulation. Immune complexes, we talked about how immune complexes can be a problem. They are cleared by the phagocytes that are, these complexes are tagged for phagocytosis, but if not, they can cause autoimmunity. Erythrocytes, so if erythrocytes are targeted by some of these immune complexes, like targeted by C3b, they can be targeted by those erythrocytes, carries immune complexes to the liver or spleen, where they are detached, and then those immune complexes are taken up by the macrophages. B-cell activation, we know it acts as this CR2 acts as a co-receptor for B-cell activation. We looked at this last time. Complement deficiencies, defect in early complement pathway, C2, C1q, C4, all of that can cause frequent infections along with autoimmunity. Remember those immune complexes that cause the autoimmunity. It can also present with lupus. Defect in terminal components like C5, 6, 7, 8, 9 causes terminal complement defect that is susceptible to encapsulated organism infections, including disseminated Neisseria infections. Mutation in MBL pathway or like MBL gene can lead to this immune issue. However, there are a lot of people who walk around with abnormal MBL and don't have infections. So it's a little controversial whether that's significant or not. And then propyridine, factor D, C3, these are alternative pathway defects. If those are defective, propyridine is X-linked. All of these can or any other defect in alternative pathway like C3 and factor D, they can lead to pyogenic bacterial infections. What happens when regulatory proteins are defective? Factor H, again, it causes glomerulonephritis or atypical HUS. SNP and factor H can also lead to age-related macular degeneration. Factor I, we talked about. Factor I also causes atypical HUS along with frequent infections. Complement receptor defects, lack of CR3, CR4, that's because of defect in CD18 or beta chain of that integrin. That leads to defect in CR3, CR4, that's called leukocyte adhesion defect. Talked about the immune complex issues. Yeah, so not much to add. That can lead to vasculitis, immune complex glomerulonephritis, can lead to, because of glomerulonephritis is caused because of destruction of glomerular vessel walls leading to thrombosis, ischemia of the tissues and scarring. But complement system can be evaded by microbes, which means the microbes become smarter and they try to escape the complement system. Such as some fungi and gram-positive bacteria will have thick cell walls that complement cannot target. They can recruit some host complement regulatory proteins in their favor. So they'll recruit factor H and help in displacing the C3b from BB. One of the examples for this is HIV virus that expresses GP41. HIV incorporates some of the complement regulatory proteins like DAF and CD59, which kind of can help them in their pathogenesis. Or some of the microbes can mimic the complement proteins, like E. coli can use C1q binding protein like complex. Staph aureus has staph complement inhibitor, so it inhibits C3 convertase. HSV has glycoprotein C1, it prevents C3b from binding to properdin. Trypanosoma cruzi, it binds to C3b and again helps prevents the formation of C3 convertase. Vaccinia virus structurally resembles C4BP or C4 binding protein, binds to C4b and C3b and accelerates the decay of the C3 and C5 convertases. So they can actually use some of their proteins to mimic the complement function and or complement regulatory function and take care of that complement in their favor. Pseudomonas can degrade C1q using alkaline protease. Serratia can cleave C5a using its protease. Staph aureus has this staph protein A that's present in cell wall and then can bind to IgG blocking complement activation. Staph aureus also has CHIPS or chemokine inhibitory protein of staphylococci. It's an antagonist of C5a. All right, so that's the end of it. Let's look at these questions. Which of the following function of antibody does not require Fc receptor for IgG? Anyone?
Is this one E?
That's correct. IgE, because it does not require Fc, it requires FAB. Children with C3 deficiency are at high risk for bacterial fungal infections. Which of the following explains the importance of C3 in defense against microbes?
So C3 is in both the classical and alternative pathways, and it's with phagocytosis of microbes. So would this one be A?
That's correct. It's correct reasoning. All of the following are accurate statements about neonatal immunity except.
I can take this one. So going through the choices, transfer maternal IgG across the placenta is mediated by an Fc receptor structurally similar to class 1 MHC. It is an Fc receptor, but I'm not sure if it's structurally similar to class 1 MHC. Choice B, IgA is absorbed in the gut from breast milk and resecreted by the infant into the bronchial mucosa. I'm not sure if that's true. Choice C, IgA secretion into breast milk involves transport through breast epithelial cells and is dependent on the poly-IgA receptor. Choice D, transport of IgG across the neonatal intestinal epithelium is mediated by an Fc receptor structurally similar to Class 1 MHC. Choice E, loss of maternal antibodies is partly responsible for increased frequency of infections at about 6 months of age. So E seems true.
Okay.
A seems, I guess the neonatal Fc receptor we talked about is structurally similar to class 1 MHC, so that A makes sense.
Right.
And then D makes sense.
So yeah, similar to MNC class 1.
Yeah,
Okay.
exactly. And
So we have
then
B
kind
and
of
C.
between B and C, yeah. So B seems, I don't know, B seems like a long shot that your IgA is absorbed in the gut from breast milk and resecreted by the infant into the bronchium. So that one would be, yeah.
Okay, sounds good. Yeah, so C sounds right. It actually is secreted in breast milk, and it uses a poly-Ig receptor. All right, last one, 15-year-old boy, multiple episodes of severe abdominal pain, intravascular hemolysis, hemoglobinuria, and looks like a diagnosis of PNH was made. What's the basis of loss of RBCs in this patient?
So is involved the DK accelerating factor. So choice C.
The accelerating factor is correct. Yeah, so that's defective. That leads to PNH.